Hello, everyone, and thank you for this opportunity to share our research with scientific community. And the topic of this lecture is, as already mentioned, is mass transfer to the volume of the gel from the system of artificial microchannels. So our research is related to bioprinting industry, and bioprinting in its essence is a task of composing cells in a particular order and ensure their proper growth so they can form a biological tissue or an organ. For this purpose, cells are immersed in special materials like gels, and the combination of cells and such medium is called bioink. After cells are composed in a particular order using bioprinter, a new challenge arises: how to ensure cells' proper growth so they can form a tissue. Proper conditions are necessary for cells development, such as nutrients and oxygen, and also products of cells' metabolism must be removed from cells. A potential solution is to build artificial network of channels similar to vascular system that can be designed in order to deliver nutrients and remove products of metabolism from cells that are immersed in gel sample. But what medium can we use for storing cells? As we found in our research, hydrogels can be considered as promising materials to solve this problem. It has certain advantages such as technological suitability for bioprinting, sufficient mechanical strength, high internal mass conductivity, biological compatibility, bioresorbability. So when cells are growing, the material can be naturally removed, and then we only get the final product as a tissue or an organ. So as we found, hydrogels can be used for these applications. What is a gel in its essence? So basically, gel is a dispersed system of high molecular structure filled with liquid. Actually, about 98% of the structure is liquid. Also, gels can be characterized by fluidity under certain conditions, also elasticity, thixotropy, mass conductivity, and also it's important to know that gels are non-stationary and non-uniform systems. Since gel can be considered as a medium for storing cells, it's important to study mass transfer properties in order to understand how to ensure reliable delivery of nutrients to cells. And mass transfer in cells occurs due to diffusion, and in order to study diffusion phenomena in gel, a color liquid can be used as presented on a picture on the right. What is the best way to study how nutrients can be delivered to cells through gel? We can study real-life systems that includes stem cells, blood substitutes, but it will require a lot of resources and time. So we can choose to use modeling systems, such as yeast cells instead of stem cells, gels, simple gels like agarose, and simple nutrients like broth. And such simple modeling systems can perform well in experiments and allow us to conduct a lot of experiments in a short period of time. Also, remote optical methods are convenient for studying cells in gels because there is no mechanical intervention in gel samples. Here you can see a scheme of optical experimental setup with two working areas. Also, there is a spectrometer, a camera, and a laptop for data analysis. Our first experiment was to understand how cells are developing in a gel sample without any sources of nutrients and oxygen. And we found that there are 4 distinctive phases, so basically 4 stages. First is a lag stage, when cells are adapting to a new environment and they're not dividing. Then it followed by second stage of exponential growth, when cells are actively growing and dividing. And then there is a stationary phase, when the population of cells remain relatively stable. And finally, extinction phase, so the population of cells is reducing due to lack of nutrients and oxygen. The final stage, the 4th stage, is not desirable for bioprinting applications, because we want to keep cells at stage 3, so we can maintain the same amount of cells for long period of time, so they can form a tissue. So we need to study how to effectively deliver nutrients and oxygen to cells, so they can grow and divide for a long period of time. So the first idea was to deliver nutrients using diffusion phenomena, as depicted on the picture on the left. So we used a colored nutrient, so it was more convenient to observe how it penetrates a gel sample. And as you can see, there is a non-linear dependence. So the most part of nutrients is on the top of a gel sample, and the deeper we go in a gel sample, the less nutrients go in these areas. Also, spectral characteristics were measured for these gel samples, and we can see 2 types of curves on this plot, which correspond to different concentrations of agarose gel. And using this spectral data, we can derive diffusion coefficient. So after we obtained data about diffusion coefficient, we can now get information how this how diffusion is related to concentration of agarose, and what happens if we combine agarose with different substances like starch. And we found that actually we can tune diffusion properties of gel more precisely if we can add some new substance. For example, we have experimented with combinations of agarose gel and starch, and found that a small portion of starch can change diffusion properties more precisely. Intensity of light transmission also was studied in relation to different levels of gel sample. And in the upper level, there is a nutrients liquid. Then there is a border between nutrients and gel. And also area where yeast cells are located. In case of small time scale, about 15 minutes, light transmission decreases on the border between liquid and gel. And after 24 hours, light transmission in gel becomes lower due to cells division. So as we can see on the picture, a second curve is after 24 hours, and first data points. First data points after 15 minutes. Second set of data points is after 24 hours. So light transmission in gel becomes lower, and because there are more cells after 24 hours, and Also, we can see significant drop in light transmission on a border between gel and nutrient liquid, which can be explained by the fact that after 24 hours, a solid border between nutrient liquid and gel was formed. Behavior of nutrient and cells concentrations were studied at two different time scales, as was mentioned before, namely 15 minutes and 24 hours. So at small time scale, cell division is negligible and cells number of, or basically concentration of cells remain constant. At the same time, nutrient concentration decreases by have non-linear characteristics. If we look at the same dependence after 24 hours, we can see that both cells concentration and nutrient concentration decreases by in a non-linear manner. So since it's difficult to penetrate to deep layers of gel, so it's hard to deliver nutrients and oxygen in the deeper layers, an artificial channel can be formed in gel to intensify nutrients delivery. As you can see on a picture, a colored nutrient was delivered through artificial channel, and over the time it penetrates the borders of a channel and fill a gel sample. Also, in case of complex structures, we can form branched channels. So in this case, we can build a root channel and then develop branched channels. In this case, the whole gel sample can be filled with nutrients and oxygen more uniformly. Also, in order to compare performance, a gel sample with two channels and one channel was studied. And as you can see from the picture, part of a gel with two channels demonstrate more intense filling of gels of nutrients in a sample compared to the part of gel with only one channel. So it's more beneficial to have a wide network of channels inside gel to ensure proper nutrition of cells. Also, a four-layered gel sample was composed with alternating layers containing cells and without cells. In such way, cells can be isolated from each other. This can be useful, for example, if we can form different types of tissues in the same area. So we can isolate them with a gel that have high diffusion coefficient, and it would be difficult for cells from one area to move to another. And as we can see on other slide, when we look at population of cells, we see that they remain constant in a gel sample where they were immersed, but then when there is a border between one layer of gel and another with higher density, we see a drastic decrease in cells population. So as we can see, such layers can isolate one area with cells from another. Also, in addition to experiments, a mathematical model was developed. It takes into account nutrients concentration, cells concentration, and the intensity of nutrient consumption by cells. Also, model considers a lag phase. Basically, it's a time delay before cells were immersed in gel and time when they start actively growing and dividing. So basically, this part of a model. And here we can see curves which were plotted based on this model, and there are three distinctive stages on this plot. So first corresponds to lag phase. So the number of cells remain constant, and the amount of nutrient is constantly growing because the demand for nutrients from cells is not changing. Then we move to the second phase, where cells start actively growing and dividing, and consumption of nutrients increase. And then we finally move to a final stage where nutrient concentration and cells concentration find the equilibrium. So based on above mentioned results, the following conclusions can be made. The growth of microorganism in gel is localized near the surface, and the width of this zone can be estimated from 3 to 4 millimeters. A system of artificial channels created in gel can be used for delivering nutrients to cells in the deeper layers of gel. Channels remain stable over a long period of time, and hence can be reliable methods for nutrients delivery for a long time as well. Also, usage of optical methods based on the light absorption intensity measurement can be used for monitoring cells growth. And also the possibility of cells growth over the whole volume of gel was shown. Thank you for your attention. If you have any questions, I'm ready to answer.